Good morning. Dear students, today you are going to learn about cholesterol metabolism. So, this cholesterol metabolism consists of three important things. One is de novo synthesis of cholesterol and its regulation. Then the second one is transport of cholesterol. And third one is degradation or the important substances that are formed from the cholesterol. So, let us learn about them today in detail. The learning objectives of today's topic are: describe metabolism of cholesterol, its significance and regulation. De novo synthesis of cholesterol, its significance and regulation. Metabolic fates of cholesterol, its significance and regulation. The cholesterol metabolism. The cholesterol is a major sterol in the humans. Cholesterol is an amphipathic lipid which can be synthesized by most cells of the body and is obtained from the diet in the food of the animal origin. So that means what? The cholesterol has got two sources. One is endogenous source where the body has the capability to synthesize the cholesterol within the body, and second one is exogenous source, that the cholesterol that is obtained from the diet. So the two sources of cholesterol to the body, and it is not synthesized in the plants. The major source of dietary cholesterol is egg yolk and meat, and in the meat, particularly the organ meat like liver. Any an abnormality in either cholesterol metabolism or transport through the plasma appears to be related to the development of atherosclerosis that can lead to myocardial infarction or stroke. So these are the topics, that is atherosclerosis is the topic that you are going to learn in detail in the coming classes. So you remember that any abnormality in the cholesterol metabolism or transport is related to the development of atherosclerosis and which can lead to myocardial infarction and stroke. Then the structure of cholesterol. So cholesterol is a 27 carbon compound which consists of cyclopentanoperhydrophenanthrene nucleus. So here in the diagram you can see that, so A, B, C, D. So this is the cyclopentanoperhydrophenanthrene nucleus which consists of 19 carbons. And in this 19 carbons, at the third carbon, there is hydroxyl group is attached to it, and a double bond is present between the fifth and sixth carbon, and a 8 carbon side chain is attached at the 17th carbon. So this makes the cholesterol a 27 carbon compound. So cholesterol, it is a 27 carbon compound containing cyclopentanoperhydrophenanthrene nucleus of 19 carbons. So where on one hydroxyl group at third position, which is the characteristic of all sterol, is present. The hydroxyl group is beta oriented, projecting above the plane of the ring, and a double bond is present between the carbon atoms 5, 5th and 6th, and the 8 carbon side chain, which is beta oriented, is attached at 17th carbon. So this makes the cholesterol a 27 carbon structure. So this is the chemistry and structure of cholesterol. The major sites for the cholesterol biosynthesis are liver, adrenal cortex, testis, ovaries, and intestine. So all the nucleated cells can synthesize cholesterol, including arterial wall, but the enzymes for this cholesterol biosynthesis are partly located in the endoplasmic reticulum and partly in the cytosol. The requirements for the cholesterol biosynthesis. The acetyl CoA is a 2 carbon compound which provides all the 27 carbon atoms for the cholesterol biosynthesis. The reducing equivalents are supplied by NADP H plus H plus, and the energy for the synthesis of cholesterol is provided by ATP. For production of one molecule of cholesterol, there are 18 moles of acetyl CoA are required, and 36 moles of ATP and 16 moles of NADP H plus are required for the synthesis of cholesterol, one molecule of cholesterol. Then de novo synthesis of cholesterol. Cholesterol is synthesized by most cells of the body, particularly liver and intestine are the major sites for the cholesterol biosynthesis. All those 27 carbon atoms of the cholesterol are derived from a 2-carbon compound, that is acetyl CoA. The reactions for the cholesterol biosynthesis occurs in 5 stages. So let us learn about these 5 stages in detail. So synthesis of mevalonate from acetyl CoA is the first stage, and synthesis of isoprene units from the mevalonate is the second stage, and synthesis of squalane from isoprene unit is a third stage, and synthesis of lanosterol, which is the first sterol formed in the cholesterol biosynthesis, is the fourth stage in the cholesterol biosynthesis, and formation of cholesterol from the lanosterol is the fifth and final stage for the cholesterol biosynthesis. So the first 2 stages takes place in the cytoplasm, that is formation of mevalonate and isoprene units occurs in cytoplasm, and next 3 stages, that is formation of squalane, lanosterol and cholesterol occurs in endoplasmic reticulum. The first stage is the condensation of 3 molecules of acetyl CoA to form mevalonate, and the second stage is conversion of mevalonate to the activated isoprene units, and third stage is the polymerization of 6 isoprene units to form squalane. And the fourth stage is cyclization of squalane to form a parent steroid nucleus, that is lanosterol. It is the first formed sterol in the cholesterol biosynthesis, and fifth and final stage is the formation of cholesterol from lanosterol. So the biosynthesis of cholesterol showing its 5 stages. The 2 molecules of acetyl CoA condenses to form acetoacetyl CoA in presence of enzyme thiolase with the liberation of CoAsh. So another molecule of acetyl CoA condenses with acetoacetyl CoA in presence of enzyme HMG CoA synthase to form HMG CoA, that is hydroxymethylglutaril CoA. So these reactions are similar to that of ketone body synthesis, but the ketone body synthesis occurs in mitochondria, but these reactions are occurring in the cytoplasm. That means there are 2 different pools of HMG CoA. That is, one pool of HMG CoA is present in the mitochondria, which is responsible for the synthesis of ketone bodies, and another pool of HMG CoA is present in the cytosol, which is responsible for the synthesis of cholesterol. So the 2 pathways are distinct, that is ketone bodies are produced in the mitochondria while the cholesterol synthesis occurs in the cytosol. It indicates there are 2 different pools of HMG CoA for the synthesis of ketone bodies and cholesterol present in the mitochondria and cytosol respectively. Formation of mevalonate. So HMG CoA reductase is the rate limiting enzyme in the cholesterol biosynthesis. This enzyme is present in the endoplasmic reticulum, which catalyzes the conversion of HMG CoA to mevalonate. It requires 2 molecules of NADP H+H+. So this enzyme is controlled by hormones, by certain drugs, and also by transcription and translation. So let us learn about them in detail in regulation. So HMG CoA reductase is responsible. It is the rate limiting reaction in the cholesterol biosynthesis, which is responsible for converting HMG CoA to mevalonate. Then production of isoprenoid units from mevalonate. In a 3-step reaction catalyzed by the kinase, the mevalonate is converted to 5-phosphomevalonate, and 5-phosphomevalonate is converted to 5-pyrophosphomevalonate, and 5-pyrophosphomevalonate is converted to 3-phospho-5-pyrophosphomevalonate. It requires 3 molecules of ATP. And 3-phospho-5-pyrophosphomevalonate undergoes decarboxylation to form isopentenyl pyrophosphate. It is the first isoprenoid unit formed during the cholesterol biosynthesis. So, the formation of isopentenyl pyrophosphate from mevalonate requires 3 molecules of ATP. For synthesis of 1 molecule of isopentenyl pyrophosphate, it requires 3 molecules of ATP. Remember it. So it is once there is formation of isopentenyl pyrophosphate, which is the first formed isopentenyl unit, it isomerizes to form dimethyl allyl pyrophosphate, which is also a 5 carbon compound. Then these two molecules, that is isopentenyl pyrophosphate and dimethyl allyl pyrophosphate, both are 5 carbon isopentenyl units. So here you can see how the mevalonate is converted into isopentenyl unit, where the mevalonate, in presence of enzyme mevalonate kinase, it forms 5-phosphomevalonate. It requires an ATP, that is first ATP. Then the mevalonate 5-phosphate is converted to 5-pyrophosphomevalonate, which also requires a molecule of ATP, and kinase is the enzyme. 5-pyrophosphomevalonate gets converted into 3-phospho-5-pyrophosphomevalonate. It also requires a molecule of ATP. Finally, 3-phospho-5-pyrophosphomevalonate undergoes decarboxylation in presence of enzyme decarboxylase to form isopentenyl pyrophosphate, which is the first formed isopentenyl unit in this reaction. So this isopentenyl pyrophosphate undergoes isomerization in presence of enzyme isomerase to form dimethyl allyl pyrophosphate. So this is how the 3 molecules of ATP are required for formation of 1 molecule of isopentenyl pyrophosphate from mevalonate. So these are the energetics of the cholesterol biosynthesis. So remember it that 3 ATPs are required for synthesis of 1 molecule of isopentenyl pyrophosphate. So this once the isopentenyl pyrophosphate is formed, it isomerizes to form dimethyl allyl pyrophosphate. So here the isopentenyl pyrophosphate condenses with dimethyl allyl pyrophosphate to form a 10 carbon compound, that is geranyl pyrophosphate, in presence of enzyme cis-phenyl transferase. So geranyl pyrophosphate is a 10 carbon compound. So this 10 carbon compound, that is geranyl pyrophosphate, condenses with another molecule of isopentenyl pyrophosphate to form a 15 carbon compound, that is farnesyl pyrophosphate, in presence of enzyme cis-phenyl transferase. So farnesyl pyrophosphate, which is a 15 carbon compound. So this farnesyl pyrophosphate condenses with another molecule of farnesyl pyrophosphate to form a 30 carbon compound, that is squalane, in presence of enzyme squalane synthase. This is how a 30 carbon squalane is synthesized from a 2 carbon acetyl CoA. So isopentenyl pyrophosphate and dimethyl allyl pyrophosphate condenses to produce a 10 carbon geranyl pyrophosphate. Another molecule of isopentenyl pyrophosphate condenses with geranyl pyrophosphate to form 15 carbon farnesyl pyrophosphate. 2 units of farnesyl pyrophosphate unite and get reduced to produce a 13 carbon squalane. This is how a 30 carbon squalane is synthesized from 2 carbon acetyl CoA. So conversion of squalane to cholesterol. Squalane undergoes oxidation by enzyme epoxidase using molecular oxygen and NADPH plus H plus to form squalane epoxide. Then the enzyme cyclase converts it into a 30 carbon compound, that is lanosterol. So this lanosterol is the first formed steroid compound in the biosynthesis of cholesterol. So lanosterol is the first formed steroid compound in the biosynthesis of cholesterol. The formation of cholesterol from lanosterol is a multi-step process with a series of about 19 enzymatic reactions. The most important reactions in these 19 enzymatic reactions are reducing of carbon atoms from 30 to 27, as lanosterol is a 30 carbon compound and cholesterol is a 27 carbon compound. So the important reaction is reducing the carbon atoms from 30 to 27. for formation of cholesterol from lanosterol and removal of two methyl groups. So this is done by removal of two methyl groups from C4. One methyl group is removed and from C14 to produce zymosterol. So that is removal of two methyl groups from C4 and C14 to produce zymosterol. Shift of double bonds from C8 to C5. The reduction of double bonds present between C24 and C25. The enzymes involved in conversion of lanosterol to cholesterol are associated with endoplasmic reticulum. 14 desmethyl lanosterol, zymosterol, cholesterol, desmosterol are among the important intermediates in the cholesterol biosynthesis. The penultimate product is 7-dehydrocholesterol, which on reduction finally yields cholesterol. So here is the summary of the cholesterol biosynthesis, where the two molecules of acetyl-CoA condenses in presence of enzyme thiolase with the liberation of CoA to form acetoacetyl-CoA. So this acetoacetyl-CoA condenses with another molecule of acetyl-CoA to form HMG-CoA, that is hydroxymethyl-glutyryl-CoA, with the liberation of CoA in presence of enzyme HMG-CoA synthase. So this HMG-CoA, in presence of enzyme HMG-CoA reductase, it requires two molecules of NADPH plus H plus to form mevalonate. So this is the regulatory step in the cholesterol biosynthesis, and HMG-CoA reductase is the regulatory enzyme in the cholesterol biosynthesis, which is regulated by drugs, by hormones, and by transcription, translation, and also by feedback regulation. So let us learn about it in detail. So in the next slide, so here you understand that this is the important step in the cholesterol biosynthesis, where the mevalonate is synthesized from HMG-CoA by the enzyme HMG-CoA reductase. So mevalonate in presence of enzyme kinase undergoes phosphorylation to form 5-phosphomevalonate, and 5-phosphomevalonate undergoes phosphorylation to form 5-pyrophosphomevalonate, and 5-pyrophosphomevalonate in presence of enzyme kinase undergoes phosphorylation to form 3-phospho-5-pyrophosphomevalonate. So this 3-phospho-5-pyrophosphomevalonate undergoes decarboxylation in presence of enzyme decarboxylase to form isopentenyl pyrophosphate. So this is the first formed isopenide unit in the cholesterol biosynthesis. It is a 5-carbon compound. So isopentenyl pyrophosphate isomerizes to form dimethyl allyl pyrophosphate. So this isopentenyl pyrophosphate condenses with dimethyl allyl pyrophosphate to form a 10-carbon compound, that is geranyl pyrophosphate, in presence of enzyme cis-phenyl transferase. So this geranyl pyrophosphate, which is a 10-carbon compound, condenses with another molecule of isopentenyl pyrophosphate to form a 15-carbon compound, that is farnesyl pyrophosphate, in presence of enzyme cis-phenyl transferase with the liberation of pyrophosphate. So the farnesyl pyrophosphate, which is a 15-carbon compound, condenses with another molecule of farnesyl pyrophosphate to form a 30-carbon compound, that is squalane. In presence of enzyme squalane synthase, it requires a molecule of NADPH plus H plus. So this is how a 30-carbon compound squalane is synthesized from a 2-carbon compound acetyl-CoA. So once the squalane is synthesized, then the lanosterol is synthesized from the squalane by the enzymes like epoxidase, hydroxylase, and cyclase. So lanosterol is the first formed sterol in the cholesterol biosynthesis, and the lanosterol undergoes a series of 19 reactions to form cholesterol, which is a 27-carbon compound. This is how the cholesterol is synthesized from acetyl-CoA. Then energy cost for cholesterol synthesis. Cholesterol biosynthesis is a complex and energy expensive process. The ATP is consumed only in the step that converts mevalonate to activated isoprene units. So 3 ATP molecules are used to create each of the 6 activated isoprene units required to construct squalane for a total cost of 18 ATPs. So for the synthesis of one molecule of cholesterol, 18 molecules of ATP is required. So synthesis of cholesterol is an energy consuming process which requires 18 molecules of ATP for synthesis of one molecule of cholesterol. Then regulation of de novo synthesis of cholesterol. Cholesterol biosynthesis is a complex and energy expensive process. Excess cholesterol cannot be catabolized for the use as a fuel and must be excreted. In mammals, the cholesterol production is regulated by intracellular cholesterol concentration, supply of ATP, and hormones glucagon and insulin levels. So regulation of de novo synthesis of the cholesterol is affected by three factors. So is influenced by three factors, that is short term and long term regulation, that is hormonal regulation and regulation by drugs. So let us learn about them. The synthesis of mevalonate from HMG CoA reductase is the committed step in the cholesterol biosynthesis. In the short term, regulation of the activity of existing HMG CoA reductase is regulated by reversible covalent alteration, that by phosphorylation and dephosphorylation. So the hormone glucagon stimulates its phosphorylation. So hormone glucagon phosphorylates the enzyme HMG CoA reductase and inactivates it, and thereby decreases the cholesterol synthesis. And hormone insulin promotes dephosphorylation of HMG CoA reductase and activates it by dephosphorylation and is favoring the cholesterol synthesis. So this is how the hormone insulin and glucagon, they activate and deactivate the enzyme HMG CoA reductase by phosphorylating and dephosphorylating it and regulating the cholesterol biosynthesis. In long-term regulation, the number of molecules of HMG CoA reductase is increased or decreased in response to cellular concentrations of cholesterol. The HMG CoA reductase in the liver is inhibited by mevalonate and by cholesterol. The mevalonate and cholesterol repress transcription of HMG CoA reductase via activation of transcription factor, that is sterol regulatory element binding protein, and thus decreases the cholesterol synthesis. So apart from phosphorylating and dephosphorylating the enzyme, the HMG CoA reductase, so the mevalonate and cholesterol, which are the products formed in the cholesterol biosynthesis, they repress the transcription. So they regulate the activity of enzyme HMG CoA reductase by transcription and translation, in short. So they repress the transcription of the HMG CoA reductase via activation of transcription factor, that is sterol regulatory element binding protein, and thus decreases the cholesterol biosynthesis by regulating the transcription and translation, that is synthesis of enzyme HMG CoA reductase. The small quantities of oxysterol such as 25-hydroxycholesterol are formed in the liver and acts as regulators of cholesterol synthesis. The oxysterols inhibit the cholesterol synthesis by stimulating proteolysis of HMG CoA reductase. Then regulation by drugs. Levostatin and other statin group drugs are competitive inhibitors of HMG CoA reductase. These drugs are used to reduce the cholesterol levels in the blood. So these drugs, that is statins, they act as the competitive inhibitors of HMG CoA reductase. So they inhibit the activity of the enzyme by acting as competitive inhibitors and regulate the blood cholesterol levels. So here, apart from it, you can see in the diagram that how the hormones, that insulin, so it activates the enzyme HMG CoA reductase by dephosphorylating it, and converts the HMG-CoA to mevalonate. In the same way, the hormone glucagon, it inhibits the activity of enzyme HMG-CoA reductase by phosphorylating it or by inactivating it through phosphorylation and inhibiting the activity of HMG-CoA reductase. So apart from it, you can see here that the products like mevalonate and cholesterol, so they also inhibit the formation of HMG-CoA reductase by transcription and translation. So this is the total chart or a diagram where you can see how the cholesterol synthesis is regulated by regulating the enzyme, that is HMG-CoA reductase, which is a very important and regulatory enzyme in the synthesis of cholesterol. So one is, that is, it is regulated by drugs where the drugs acts as a competitive inhibitors of this enzyme, and it is regulated by hormones, so where the insulin dephosphorylate the HMG-CoA reductase and activates it and converts HMG-CoA to mevalonate, and glucagon, it phosphorylate the enzyme HMG-CoA reductase and inactivate it and inhibit the conversion of HMG-CoA to mevalonate. So this is how the hormones regulate apart from the drugs, and then the products which are formed. So like mevalonate and cholesterol, they control the synthesis of the enzyme HMG-CoA reductase by transcription and translation. So they inhibit the formation of enzyme HMG-CoA reductase if their concentration is more. So this is how the products feedback inhibition occurs, and they also contribute for the regulating of blood cholesterol levels by transcription and translation. So this is the regulation of cholesterol biosynthesis by different factors. Then transport of cholesterol. Cholesterol is transported in body fluids in the form of lipoprotein particles. Cholesterol esters, which are resynthesized in the mucosal cell together with some anesterified cholesterol, are incorporated into chylomicrons, which transport the cholesterol and other dietary lipids from the intestine. During circulation, only about 5% of cholesterol esters is lost, and rest of the 95% of chylomicron cholesterol is delivered to the liver through LRP receptors. LRP receptors are nothing but LDL receptor related proteins. So they are delivered to these LRP receptors in the form of chylomicron remnants. The cholesterol in excess of the liver's own need is exported in the blood in the form of VLDL, that is very low density lipoprotein. Triacylglycerols of VLDL are hydrolyzed by the action of enzyme lipoprotein lipase. The resulting remnant, which are rich in cholesterol esters, are called intermediate density lipoprotein. So the enzyme lipoprotein lipase is responsible for the synthesis of intermediate density lipoprotein from VLDL. Half of the ideal, that is intermediate density lipoprotein, are taken up by the liver for processing, and half are converted to low density lipoprotein, that is LDL, by removal of more triacylglycerol. So low density lipoprotein is a major carrier of cholesterol in the blood from the liver to the peripheral tissues. The role of LDL is to transfer the cholesterol to the peripheral tissues through LDL receptors with the help of ACAT enzyme and regulate de novo cholesterol synthesis. The HDL, that is high density lipoprotein, picks up the cholesterol from the peripheral tissues and from other lipoproteins and converts it to cholesterol esters by LCAT enzyme. The HDL cholesterol esters are ultimately returned to the liver through HDL receptors for excretion, where it is degraded or excreted into the bile. So HDL is responsible for removing of cholesterol esters from the peripheral tissues, and LDL is responsible for the deposition of the cholesterol esters to the peripheral tissues. So this is the main difference between these two lipoproteins, that is LDL is responsible for deposition of cholesterol eh, and its esters into the peripheral tissues, but whereas HDL acts opposite to it and it removes the cholesterol esters from the peripheral tissues and hand it over to the liver. So good cholesterol and bad cholesterol. HDL cholesterol is considered to be the good cholesterol because it acts as a scavenger, as it scavenges the excess cholesterol from peripheral tissues and transport it to the liver where it is degraded or excreted in the bile. HDL thus tends to contribute for low blood cholesterol levels. On the other hand, LDL cholesterol is called bad cholesterol because it transports cholesterol from the liver to the peripheral tissues and is responsible for the deposition of cholesterol in the peripheral tissues, which may be a reason for atherosclerosis, hypertension, myocardial infarction, or stroke. Hence, a positive correlation exists between the incidence of the coronary atherosclerosis and blood circulation of LDL cholesterol. Hence, LDL cholesterol is considered as a bad cholesterol, and HDL cholesterol is considered as a good cholesterol because it scavenges the excess cholesterol from the peripheral tissues and transports it to the liver. So it is responsible for the decrease of cholesterol levels at the peripheral tissues. Hence, it is considered HDL is considered to be a good cholesterol. But whereas LDL cholesterol is responsible for deposition of cholesterol into the peripheral tissues or transport the cholesterol from the liver to the peripheral tissues and is the main reason for atherosclerosis, myocardial infarction, coronary atherosclerosis, and stroke. Hence, it is considered as a bad cholesterol. Then, metabolic fates of cholesterol. Then, functions of cholesterol. Cholesterol is the most abundant sterol in the humans and perform a number of essential functions. For example, it is a major constituent of plasma membrane and plasma lipoproteins. It is a precursor for bile salts. It is a precursor of steroid hormones that include adrenocortical hormones, sex hormones, placental hormones, etc. It is also a precursor of vitamin D, cardiac glycosides, cystosterols of the plant kingdom, and some alkaloids. It is required for nerve transmission. Cholesterol is widely distributed in all cells of the body, but particularly abundant in the nervous tissue. Metabolic fates of cholesterol. The cholesterol has several metabolic fates. A small fraction of cholesterol made in the liver is incorporated into the membranes of the hepatocytes. A small quantity of oxysterol, such as 25 hydroxycholesterols, are formed in the liver and acts as the regulator of the cholesterol synthesis. The oxysterols inhibit cholesterol synthesis by stimulating proteolysis or breakdown of the enzyme HMG-CoA reductase, thereby inhibiting the cholesterol biosynthesis. So, in other tissues, the cholesterol converted into steroid hormones in the adrenal cortex and gonads. Vitamin D. So, vitamin D is considered as a hormone. So, no other vitamin is considered as a hormone, but vitamin D is considered as a hormone because it functions like a hormone. So, it has got some target organs to act specifically, like the hormones have the target organs. So, that's why vitamin D is considered as a hormone. So, vitamin D hormone or vitamin D is synthesized in the liver and kidney, which regulates the calcium and phosphorus metabolism. So, which you have learned about it in detail during the fat soluble vitamins. So, most of the cholesterol is exported in one of the three forms as bile acids, biliary cholesterol, or cholesterol esters. Then, synthesis of bile acids and its regulation. So, cholesterol is converted to 7 alpha hydroxycholesterol in presence of enzyme 7 alpha hydroxylase. So, from this 7 alpha hydroxycholesterol, so the bile acids are synthesized. So, the primary bile acids are cholic acid and chenodeoxycholic acid. So, they both conjugate with glycine and taurine amino acid to form glycocholic acid, taurocholic acid, glycokenodeoxycholic acid, taurokenodeoxycholic acid. So, these four are the primary bile acids. So, when they get conjugated with sodium and potassium, they form the bile salts. So when these bile salts enters into the intestine, they are acted upon by the intestinal bacteria and they get converted into secondary bile acid. So they are deoxycholic acid and lithocholic acid. So these are the secondary bile acids. So the cholesterol is responsible for the formation of bile acids and bile salts. So these bile acids which are formed, so they regulate the synthesis of bile acids and bile salts by feedback inhibition or feedback regulation by regulating the enzyme 7 alpha hydroxylase. So cholesterol activates the enzyme 7 alpha hydroxylase, and excess of bile acids and bile salt they inhibit the enzyme 7 alpha hydroxylase. This is how the bile acids and bile salts are synthesized and regulated from cholesterol. So catabolism of cholesterol. The major pathway for catabolism of cholesterol is its conversion into bile acids and bile salts in the liver. The cholesterol is first converted into 7 alpha hydroxycholesterol by 7 alpha hydroxylase. So 7 alpha hydroxylase is a part of microsomal hydroxylase system. The most of the 7 alpha hydroxycholesterol is converted into cholic acid. The cholic acid can combine with glycine and taurine to form glycocholic acid and taurocholic acid respectively. Some of the 7 alpha hydroxycholesterol is also converted to quinodeoxycholic acid. So this can combine with glycine to form glycoquinodeoxycholic acid, and it can combine with taurine to form tauroquinodeoxycholic acid. So the cholic acid and quinodeoxycholic acid are the first formed bile acid from 7 alpha hydroxycholesterol. So which conjugate with glycine and taurine to form glycocholic acid, taurocholic acid, glycoquinodeoxycholic acid, and tauroquinodeoxycholic acid. So glycocholic acid, taurocholic acid, glycoquinodeoxycholic acid, and tauroquinodeoxycholic acid are the primary bile acids. Their sodium and potassium salts are known as bile salts. Bile salts are formed in the liver and are excreted through bile in the intestine. Most of the bile salts entering the intestine are reabsorbed into the portal blood. These are brought to the liver and are re-excreted into the intestine by the liver. This is known as enterohepatic circulation of bile salts. Intestinal bacteria convert the unabsorbed bile salts into secondary bile acids and sterol. So, deoxycholic acid and lithocholic acid are the main secondary bile acids. Coprostanol is the main sterol formed from the bile salts. So, these are excreted in feces. So, this is about the formation of bile acids and bile salts from the cholesterol. Then excretion of cholesterol. Average diet contains about 300 milligrams of cholesterol per day. The body synthesizes about 700 milligrams of cholesterol per day. About 500 milligrams of cholesterol is excreted through bile. Some of this partly reabsorbed from the intestine and remaining is converted into bile acids which are excreted in the bile as bile salts. The cholesterol is excreted in feces. Unlike many other metabolites, the cholesterol cannot be destroyed by oxidation to carbon dioxide and water because of the absence of enzymes capable of catabolizing the steroid ring of the cholesterol. It is excreted in the bile either as cholesterol or after conversion to bile acids. So this is the assignment on cholesterol metabolism. So you need to complete this assignment on cholesterol metabolism and send it back to me on Telegram.